you should hopefully be able to see kind of a striated appearance, a striped appearance under the microscope. So both skeletal muscle and cardiac muscle is striated. So it has this kind of stripiness down the microscope. So I'm just going to write some keywords in here. Also, when you look at skeletal muscle under a microscope, it's going to have these long, straight muscle fibres. So hopefully it should kind of be parallel to each other and have these long fibres running down, like what's in here. It's kind of running lengthways. So it should be, fingers crossed, kind of these long, straight fibres. So we say it's unbranched. Another characteristic feature when you look at skeletal muscle is the nuclei. So the nuclei, some people won't be able to see it, but vast majority again should. And if you use some generic stain, the skeletal muscles stain like a pale pink colour, and the nuclei look a lot more dark blue or purple. And in skeletal muscle cell, it's got lots of nuclei, so it's said to be multi-nucleated. So you've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, just on that muscle fibre there. So it's got many nuclei, it's multi-nucleated. And the nuclei are located on the edge of the muscle fibre. So you should, fingers crossed, see them towards the edges rather than in the middle. So it's said to have peripherally, or if you don't want to remember the word peripherally, it's just at the edge. So peripherally located nuclei. So they're at the edge. Oh, so I spelled that wrong, Anna. Go on, how do you spell it? Let's go back. P-R-I-P-H-E rally. I'll leave the writing on while you're working so you have got the writing in. The last thing kind of about muscle fibres for skeletal muscle is quite big. They're around 10 to kind of 100 micrometers. So they're bigger than cardiac muscle and they're bigger than the smooth muscle. So around 10 to 100 micrometers. So a micrometer is 1,000th of a millimetre.
That's a nuclei at bottom.
Nuclei at the bottom.
Do you know that it's a striated, is that how you say it?
Yeah.
Muscle. What's that actually mean?
It means it's got a striped appearance. So the muscles are made up of proteins, and the proteins are called actin and myosin, which we'll look at under the electron microscope. But it just means it's got kind of that striped pattern to it.
Yeah, okay.
So that's just kind of the image I showed you before. So you can see that long, straight fibre, the nuclei located on the edge of that fibre, and you'll see on high power that stripe pattern, hopefully. So that's what you're looking for with skeletal muscle. They're the characteristic features. So when your kind of learning outcome says describe the microscopic structure of striated muscle, that's kind of the general features you'll be looking at that under a microscope. So you can kind of list those things, describe those things in a diagram. So this kind of slightly higher magnification what you'd get. It's not much higher. You get about 400 times. This is probably around 800 times, 1,000 times. So it's a little bit more zoomed in than what you'll see. But you can see that's a very, very nice striped banding pattern. You've got, again, the nuclei at the edge of the skeletal muscle cell, and lots of those nuclei, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So that's a characteristic skeletal muscle slide. If you were to draw that, when you're drawing these down a microscope, you're going to do this in biology too with grades when you do biology practical skills. So this is a very, very, very quick introduction. But you draw it in a pencil and ruler as big as you can single clear continuous lines. So don't kind of shade, don't colour in, don't sketch it. It wants to be single clear kind of continuous lines. So you draw it, kind of I'm drawing that one, something like that. If there's a darker area, say the nuclei are slightly kind of darker, you can do lines on them to show the darker, but you don't colour it in, you just kind of line it in or dot, do it all dotted, again just to show it's a darker area. So that's probably how you draw that one. You've got two big ones in the centre, it's a slightly darker area and that's slightly darker. So that would be kind of a typical drawing for a striated muscle cell. You might show it's got that striated appearance by putting some stripes in it. You tend not to do that through the whole thing, but just do a section to show you that it's got that striped appearance. But you'll do with Grace, when you do biology practical skills, you'll get lots of different tissues, some nervous tissue, muscle tissue, smooth muscle, fat tissue, different types of connective tissue, and you'll do a portfolio of drawings drawn in the proper scientific way. But this is just kind of an introduction to that. So if you kind of go to a diagram, this will show you how it takes that drawing and creates a diagram. So that is what you kind of draw. So Grace would probably, if you can, some people, it's really, really fiddly doing it, but sometimes you can take photos down the microscope. So she might ask you to kind of take a photo of the slide you're looking at and then stick it next to the drawing on the next page and show how you've drawn it.
Are all your slides on Moodle now?
So that's your skeletal muscle, and that's the only one we can care about at the minute. But if you want to look at cardiac and smooth, just to compare them and look at the differences. Cardiac, we do when we do cardiovascular system, so we'll do that at the end of biology 3, so kind of around April, May time. So cardiac muscle is also striated, but it has some main differences. This time it has a nuclei in the centre as opposed to the edge, and the biggest thing with cardiac is it's branched. So this is kind of showing cardiac here, kind of low medium power, so that's high power, this is low medium-ish. And you can see cardiac's got this branch structure. So we'll look at this and you have to describe this when we do heart. But you can see how it differs from the really straight skeletal. Instead it has those branches. And the nuclei tend to be more centrally located and fewer of them. So let's go back. So that's kind of showing cardiac, showing this striations. It's still striped. It's got the nuclei in the centre. And if that went to a diagram, showing those cardiac cells with a centrally located nuclei. So you can just about see the nuclei there, and the cell going down, the cell going across, the cell going there, and see how it translates. Smooth muscle doesn't have striations, so it's smooth because it's not striped, it's kind of a smooth appearance. So if you're looking at smooth muscle, centrally located nuclei, like mononucleus, it tends to have one nucleus, and it's this spindle shape to the cell. So it's kind of this elongated spindle kind of shape. So if you draw that as a diagram, kind of see the spindles forming kind of there. So again, it's slightly different structure. So you're only interested in skeletal, but it might be worth getting the other two just to see how different they look. So just kind of going back to smooth. There, you've got a smooth muscle fibre. So I'm just going to steal your book again. So next task, I've done your little diagram at the bottom so you know hopefully what you should see down the microscope on high power. You've got a box in which you can have a go at drawing it. So just single, straight, clear, continuous lines. And then some key features, which is the important bit. So I've just put 5 bullet points. As long as you've got 3 or 4 of those, that's the main thing. So if you have a go at filling that in, I'll put the words back on the board and I'm going to grab you the slides. one thing before you start. Sorry. When you're getting the slides out of the tray, the striated muscle is what your skeletal muscle is. So if the slide is written on it striated, that means skeletal. Cardiac is obviously the heart, and non-striated will be the smooth. So that's how the slide's labelled up. So they won't have skeletal written on the slide, instead it'll be called striated. Secondly, the slides you might see longitudinal LS, longitudinal section, or TS, trans-section or cross-section. So longitudinal section is if you've got your muscle, you slice the segment that way down, so it's kind of a length of the muscle, which is what you want to be looking at. You want to be kind of looking at this section sliced across, so you can see those fibres. If you get a TS, a trans-section, that's kind of cross-section, so it's you slice the muscle that way. So you kind of see fibres like that, and you slice the ends, and you can see them peripherally located nuclei that way. So when you do it, I'd advise looking at an LS, a longitudinal section. You can have a look at some TSs if there is any there, but that's kind of the main one. Okay? Right, I'll scan you the slides, microscopes are there, so I'm going to go through some slides. And I'll leave the writing up for you.
Striated.